My name is Anita Grove, and I am with the Apalachicola National Estuary Research Reserve, and welcome to our monthly SciCafe. This is a platform to share information about the science that's being conducted in the Apalachicola Bay area in an informal manner, casual setting, to bring together residents and scientists to explore different topics. Our program will be about an hour in length, and there's plenty of time for questions. And today we're going to welcome Kira Allen. Kira is our 2020-22 Margaret A. Davidson Fellow, and her talk will be river flow and sea level rise impacts on the Apalachicola Bay food web. And she is with the University of Central Florida. We've enjoyed working with her, and this is going to culminate some of her postdoctoral work. So take it away, Kira.
All right, hi everyone. Thanks for having me. I'm Kira. I'm a master's student at University of Central Florida, and I am the 2020 through 2022 Margaret A. Davidson Fellow at ANERR. And today I'll be going over the research I've done for that fellowship, looking at river flow and sea level rise impacts on the Apalachicola Bay food web. Apalachicola Bay is an estuary, meaning it lies at the intersection of freshwater and saltwater habitats. Estuaries are very vulnerable to the impacts of climate change and human-induced stressors. These can include factors such as warming temperatures, sea level rise, changes in precipitation, and upstream reallocation of river flow for human consumption. Over the past several decades in estuaries around the world, there's been notable trends of warming water temperatures and rising sea level. Climate change can result in more variable precipitation patterns and increased frequency of drought events. And these drought events can lead to greater amounts of freshwater reallocation upstream to support drinking water or agricultural needs. So Apalachicola Bay is one such estuary influenced by these climate and human-induced stressors, and two that are of particular concern for the area are freshwater reduction and sea level rise. Apalachicola River is the main source of freshwater to the bay, and there was a decades-long Supreme Court legal battle over the Apalachicola Chattahoochee Flint, or ACF, river basin, which is shown in the map on the left there. And Apalachicola River is a part of that. So in that legal battle, Georgia argued for greater freshwater diversion upstream to support growing drinking water needs. And Florida argued that receiving less freshwater inflow to Apalachicola Bay was ecologically harmful to the system. The Supreme Court ended up dismissing the case in 2021, meaning Georgia could continue with their increasing freshwater withdrawals. And as many of you may be aware, reduced freshwater input was attributed as a major factor in the decline of Apalachicola Bay oyster populations, which collapsed in 2012 and were put under a 5-year fishery closure in 2020. Another stressor affecting Apalachicola Bay is sea level rise. And this graph shows the trend in sea level since 1970, measured at the NOAA tide gauge in Apalachicola. So over that time, mean sea level has increased by about 0.2 meters with a linear rate of change of about 2.82 millimeters per year. As sea level rises, it brings a greater amount of salt water into Apalachicola Bay. When this is coupled with reduced freshwater inflow, this leads to a significant increase in the bay's salinity. Now, with a high likelihood of an increase in both freshwater reduction and sea level rise in Apalachicola Bay going into the future, it's important to be aware of how these changes might impact species who inhabit the system, particularly when the bay is home to a large number of commercially and recreationally important fishery species. An increase in Apalachicola Bay salinity due to reduced freshwater inflow and sea level rise impacts the species and biological productivity of the system. There's been many studies focusing on the response of oysters in particular to these environmental changes in Apalachicola Bay. Generally, an increase in salinity has been found to result in greater oyster mortality due to predation, disease, and physiological stress. Reduced river flow has also been found to result in lower phytoplankton and zooplankton productivity and reduced fish species richness. And increasing sea levels have been associated with lower marsh productivity. And while there's been studies investigating freshwater inflow and sea level rise impacts on certain individual species or habitat types in Apalachicola Bay, there's a need for more information to better understand how the combined influence of these environmental changes can collectively impact multiple species and the overall estuarine food web. And what I mean by the food web is the interconnected network of species who inhabit the system. So this study aims to address how changes in river flow and sea level rise affect the Apalachicola Bay food web. A coupled hydrodynamic and food web model was used to simulate future changes in freshwater inflow and sea level rise in Apalachicola Bay from 2020 up until 2050. The food web response to these stressors was measured in terms of changes in species populations over space and time. Additionally, a survey of Apalachicola Bay stakeholders was conducted to assess local ecological knowledge on Apalachicola Bay species and environmental changes. So analysis of both the model simulations and survey results were tailored around how changes in river flow and sea level rise in Apalachicola Bay would impact three central topics. These were 1, environmental conditions, 2, populations of commercially and recreationally important fishery species, and 3, the broader food web and human community. Study results provide insight on the implications of future changes in freshwater inflow and sea level rise for estuarine food web dynamics and the human communities who rely on the living resources of Apalachicola Bay. So the food web model for Apalachicola Bay was developed using EcoPath with EcoSim and EcoSpace food web modeling software, which is the most widely used food web modeling software in the world. The food web model utilizes a series of differential equations pertaining to the production and consumption of species within a system, and it uses those to calculate changes in species abundances and distributions over time and space in response to environmental changes. So the data used to construct the food web model primarily came from ANERR's trawl monitoring surveys from 2000 through 2019. These data were collected at the numbered sampling sites on the map there. The purple shaded area on the map indicates the spatial domain of the food web model. The list of species in the food web model is only a subset of the total number found in Apalachicola Bay, but I based the list on the top 99% of species caught across all years of the trawl surveys. To account for other species who may be important components of the food web, but not necessarily well represented in ANERR surveys, I also supplemented the list using data from Florida Fish and Wildlife's Fisheries Independent Monitoring Surveys and previous Northern Gulf of Mexico food web modeling studies. Now, there's a ton of information that goes into setting up the foundation of the food web model, so I'm just going to give a pretty brief overview here. To start, we need biological information on all the species represented in the model. This includes biomass as a measure of abundance in terms of grams per meter squared. Species biomasses were calculated based on length measurements collected during ANERR's trawl monitoring surveys and using length-weight regression equations to convert lengths into weights. We also need information on species production and consumption, which were derived from ratios reported in previous food web modeling studies. Next, we use data on species diets to define the trophic relationships between different species in the model. These data were sourced from previous studies and species diet databases. Lastly, we can also represent fishing effort in the food web model. Both commercial and recreational fishing fleets were represented in this model, with commercial fishery landings being sourced from Florida Fish and Wildlife trip tickets, and recreational landings being sourced from NOAA Marine Recreational Information Program surveys. So with all of this data entered, we then have a baseline foundation for the food web model. So this is a visual visualization of the Apalachicola Bay food web as represented in the model based on all that information I just went over. So with this, each circle represents a different species or fishing fleet group, and the lines indicate the trophic interactions between the different groups. So basically who consumes who. The scale along the left side indicates trophic level. So groups closer to the top are either fishing fleets or higher level predators, while groups closer to the bottom are primary producers or low-level prey species. All of these different relationships are taken into account as we move forward in the food web model simulations and analyses. Okay, the next step in calibrating the food web model is to display changes in species biomass in response to environmental conditions over time. To do that, we need time series data of both species biomasses and environmental variables. The example I've shown here is annual average biomass for bay anchovy in the plot on the left there, and then annual average Apalachicola Bay salinity in the plot on the middle. Both of those are from 2000 to 2019, which is the full length of time for which those monitoring data were available. Biomass time series were provided for all species in the food web model based on ANERR's trawl monitoring data, and time series of salinity and temperature were based on data collected by ANERR's system-wide monitoring program. And the relationship between species biomasses and environmental conditions in the food web model is defined through the use of species response curves, which looks something like the figure on the right there. That's the salinity response curve for bay anchovy. And the way to interpret the response curve is that the span of the curve indicates the salinity tolerance range for bay anchovy, and the plateau of the curve indicates the most optimum salinity levels. Salinity levels outside of the optimal range will result in lower bay anchovy biomass. Environmental response curves relating to both salinity and temperature were created for all species in the food web model where sufficient data was available. The food web model uses all of this information to calibrate the model over time and then calculates its own estimate of species biomasses in response to environmental changes. So the last step in constructing the food web model is representing the spatial dynamics of species biomasses and environmental conditions. The spatial component of the food web model consists of a gridded base map where each cell can receive environmental input and display measures of species biomass based on species environmental tolerances. So this is an example of the gridded base map for salinity in the year 2000. And on the map, cooler colors indicate less saline areas, while warmer colors indicate more saline areas. You can see more freshwater concentrated in the area where Apalachicola River feeds into the bay. The spatial domain of the food web model, which is shown by all the colored area on the map, just pertains to the area where species monitoring data were collected across Apalachicola Bay and not the surrounding channels. So all of the spatial distributions of salinity and temperature are calculated by the hydrodynamic model, which I'll talk about next. So the hydrodynamic model for Apalachicola Bay was developed by my collaborators at Tulane University using Delft 3D modeling software. It incorporates data on depth, tides, freshwater inflow, salinity, and temperature. So this image shows the depth profile of Apalachicola Bay and the surrounding area as generated by the hydrodynamic model. Cooler colors indicate deeper water, and warmer colors indicate either land or shallow water. And like the food web model, the hydrodynamic model was calibrated using existing environmental data from 2000 to 2019. So after calibrating the hydrodynamic model to observe data, it was then used to simulate future scenarios of river flow and sea level rise from 2020 through 2049. 4 different scenarios combining either low or high river flow and sea level rise were used, which are defined in the table on the top right. Low river flow conditions were based on historically low Apalachicola river flow levels from the year 2012, and high river flow conditions were based on historically high river flow levels from 1964. Low flow conditions were meant to represent a future of continual heavy upstream freshwater withdrawals, and high flow conditions were meant to represent a future where Apalachicola river flow returned to the much higher flow levels it exhibited several decades ago. A comparison of the different monthly river flow levels between 2012 and 1964 is shown in the top left plot. These monthly flow amounts were simulated through the hydrodynamic model for each year of the future scenarios. The bottom left plot compares observed annual average Apalachicola river flow from 2000 to 2019 with the annual average flow of the low and high river flow future scenarios. Sea level rise conditions were based on the observed rate of sea level rise for Apalachicola Bay. As I mentioned earlier, sea level in Apalachicola Bay has risen by about 2.82 millimeters since 1970. This rate of change was used to represent future low sea level rise conditions, and a doubled rate of change was used to represent high sea level rise conditions. So this plot on the right shows observed annual average sea level from 2000 to 2019 compared with future annual averages between the low and high sea level rise scenarios. So the different river flow and sea level rise conditions were simulated in combination with one another for a total of 4 different future scenarios. These scenarios were run through the hydrodynamic model, which output measures of water temperature and salinity in response to the changes in river flow and sea level. The output from the hydrodynamic model was used as input for the food web model, which was then able to display changes in species biomasses over time and space in response to the environmental conditions. So with the study, I wanted to not only look at the modeled simulations of the Apalachicola Bay food web, but also the knowledge and perceptions of stakeholders on how changes in river flow and sea level rise impact the system. I conducted an electronic survey of Apalachicola Bay stakeholders aimed at assessing which species stakeholders considered commercially or recreationally important and how changes in river flow and sea level rise might affect environmental conditions, fishery species, and the human community of Apalachicola Bay. A stakeholder was defined as anyone who has lived or done work in the Apalachicola Bay system. The graph on the right is a breakdown of the different types of stakeholders who participated in the survey. You can see the majority of which were local community members. So the results of both the stakeholder survey and modeled simulations offered a comparison of different knowledge types to assess how changes in river flow and sea level rise affect the Apalachicola Bay food web. As I mentioned before, analysis of these results focused on the impacts to environmental conditions, commercially and recreationally important species, and the broader food web and human community of Apalachicola Bay. I first looked at the changes predicted by the models and then compared them with the impacts anticipated by stakeholders. So starting with how changes in river flow and sea level rise affect environmental conditions, I used the hydrodynamic model results to compare temperature and salinity between 2019, which was the last year of the observed time period, and 2049 for each of the future scenarios. So these plots show the annual ranges of temperature and salinity and the percent change between the annual averages of 2019 and 2049 for each scenario. The scenarios are labeled by the river flow conditions first and then the sea level rise conditions. So if you see a label saying something like high-low, that scenario pertains to the high river flow, low sea level rise scenario. So you can see that there's an increase in annual average temperature across all future scenarios. And then annual average salinity increased during low river flow scenarios and decreased during high river flow scenarios. The hydrodynamic model also provides output of spatial distributions in temperature and salinity. So here you can see the spatial patterns between the start and end of the observed time period, so 2000 and 2019, along with the year 2049 for each of the future scenarios. For temperature, you can see there was not a lot of spatial variation in the bay during 2000 and 2019 compared with what's shown in future scenarios. During the low river flow scenarios, the southern region of the bay had much warmer temperatures, and during the high river flow scenarios, the temperatures are cooler. With salinity, we can see that the southern region of the bay is generally more saline and the northern region less saline. During the high river flow scenarios, there's a greater extent of freshwater in the bay. Now, something to note about the results for both temperature and salinity is that the changes for both the annual averages and spatial distributions are more similar between future scenarios that use the same river flow conditions than between those that use the same sea level rise conditions. So in other words, the low river flow, low sea level rise and low river flow, high sea level rise scenarios look a lot more similar than the low river flow, low sea level rise and high river flow, low sea level rise scenarios. This would indicate that Apalachicola river flow has a stronger influence on the environmental conditions in Apalachicola Bay than sea level rise. And this is a pattern that's been reported by previous studies for the region as well. So switching over to the survey results, stakeholders were asked how they thought a future reduction in river flow and future increase in sea level would impact water temperature and salinity in Apalachicola Bay. The majority of respondents thought there would be an increase in temperature due to reduced river flow and little to no change due to sea level rise. Salinity was expected to increase as a result of both reduced river flow and sea level rise. The responses to the stakeholder survey largely aligned with the results output by the hydrodynamic model, where future scenarios resulted in an increase in temperature and low river flow combined with low and high sea level rise resulted in an increase in salinity. Next, we can look at how changes in river flow and sea level rise affect commercially and recreationally important fishery species in Apalachicola Bay. Species considered to be commercially and recreationally important were determined from the stakeholder survey. Participants were given a predefined list of species to rank from most to least commercially or recreationally important, or had the option to write in other species not included in the list. So oysters are probably the most well-known commercial fishery species in Apalachicola Bay, but the aim of this study was to focus on impacts to other species, so it was noted that oysters were excluded from the list. So these plots show the results of which species outside of oysters that stakeholders considered to be the most commercially or recreationally important. The top two species in each category were selected to be used for further analysis. So for commercial fishing, this was commercial shrimp, which includes white, brown, and pink shrimp. And then the other species was blue crab. For recreational fishing, the top two species were flounders, which includes gulf and fringed flounder, and the other species was red drum, which was a write-in answer given by participants. Analysis of the food web model results focused on these 4 species groups. White shrimp was chosen to represent the commercial shrimp category, and Gulf flounder was chosen to represent the flounders category. Both juvenile and adult age classes of white shrimp were represented in the food web model, while data on the other species primarily pertain to their juvenile age class. These plots show the comparison of species biomasses between 2019 and 2049 for each of the future scenarios. So the biomasses of both juvenile and adult white shrimp increased during low river flow scenarios and decreased during high river flow scenarios. Blue crab biomass increased across all scenarios, and Gulf flounder biomass decreased across all scenarios. Red drum biomass decreased during low river flow scenarios and increased during high river flow scenarios. The food web model also gives a visualization of the spatial changes in species biomasses. So here we can see species distribution patterns between the observed time period and future scenarios. Warm colors indicate high biomass, and cool colors indicate low biomass. Biomasses of both juvenile and adult white shrimp were higher in the southern, particularly southwestern portion of Apalachicola Bay across all years and scenarios. In the high river flow scenarios, white shrimp biomass for both juveniles and adults was low across the northern, middle, and southeastern regions of the bay. Spatial variation in blue crab biomass was greater during the future scenarios compared to the observed years. And for all future scenarios, blue crab biomass was highest in the northern and middle regions of the bay. Gulf flounder biomass was generally higher in the southern region of Apalachicola Bay. And for all future scenarios, Gulf flounder biomass was low across much of the bay, except for near 2 small passes connecting to the Gulf of Mexico. Spatial variation in red drum biomass was greatest during the high river flow scenarios, and overall red drum biomass was highest in the northern region of Apalachicola Bay. So like what we saw with the environmental results, the changes in species biomasses are more similar between scenarios of the same river flow conditions than those of the same sea level rise conditions. So just as Apalachicola river flow appeared to have a strong influence on Apalachicola Bay temperature and salinity than sea level rise, the same seems to be true in regards to changes in species biomasses over time and space. So in the stakeholder survey, participants were asked how they thought a future reduction in river flow and increase in sea level would impact the selected populations of commercially or recreationally important fishery species in Apalachicola Bay. Unfortunately, there was insufficient data to examine the anticipated impacts to red drum, so analysis of the survey results focused on the impacts to commercial shrimp, blue crab, and flounders. The majority of survey respondents thought a future reduction in river flow and increase in sea level would lead to a decrease in commercial shrimp abundance. For blue crab, most respondents thought a reduction in river flow would result in a decrease in blue crab abundance, and an increase in sea level would either increase or decrease blue crab abundance in the bay. For flounders, most survey respondents thought reduced river flow and an increase in sea level would result in a decrease in flounder abundance. So comparing the survey with the food web model, we'll start with the results for white shrimp, and those were different between the survey and the model. So in the food web model, white shrimp biomass increased during scenarios of low river flow combined with low and high sea level rise, while the survey responses expected a decrease in shrimp abundance due to reduced river flow and an increase in sea level. The model results are based on the salinity and temperature tolerances of white shrimp. So looking at the response curves, we can see they tolerate a broad range of salinities and prefer warmer temperatures. The survey responses might be based on a historical decrease in shrimp fishery landings during times of drought in the system. But in these instances, there were likely other factors that contributed to this decline, such as habitat loss or an influx of new predators, which we were not able to incorporate in the food web model analysis. So looking at the results for blue crab, there were also differences between the model and survey results. The food web model showed an increase in blue crab biomass across all future scenarios, while survey responses predicted a decrease in abundance due to reduced river flow and either an increase or decrease due to sea level rise. So looking at the response curves for blue crab used in the food web model, we see they prefer low salinities and high temperatures. With a preference for low salinities, we might expect a decrease in blue crab biomass during the low river flow scenarios, but it would appear that there's still sufficient low salinity habitat areas to support higher blue crab biomass during those low river flow scenarios. The changes in blue crab abundance anticipated by stakeholders might be based on previously observed decreases in landings during drought periods, as well as trends seen in other Gulf of Mexico estuaries. For Gulf flounder, the model results did agree with those of the stakeholder survey. The food web model showed a decrease in Gulf flounder biomass across all scenarios. The response curves for Gulf flounder show that they prefer high salinities and high temperatures. The increase in salinity during low river flow scenarios actually still falls short of the optimal level for Gulf flounder, so this explains the decrease in biomass across all scenarios. The survey responses anticipated a decrease in flounder abundance due to both reduced river flow and an increase in sea level, which aligns with the results output by the food web model. To assess the impacts of changes in river flow and sea level rise on the broader Apalachicola Bay food web, I examined changes in total biomass of species in the food web model and Kempton's Q index, which is a measure of upper trophic level species diversity that can be used as a proxy for ecosystem biodiversity. Total biomass increased across all future scenarios, and upper trophic level diversity decreased across all scenarios. So looking at the spatial patterns of these variables, we see total biomass exhibited the greatest spatial variation in 2000. In 2019, total biomass was highest in the southwestern region of Apalachicola Bay, and in 2049, it was highest in the bay's northern region for all scenarios. Upper trophic level diversity had greater spatial variation in 2000 and 2019, while the spatial patterns became more uniform across the future scenarios. There's generally greater diversity in the southern region of Apalachicola Bay for all future scenarios. So the stakeholder survey was used to assess the impacts of changes in river flow and sea level rise on the human community of Apalachicola Bay. Participants were asked how a future reduction in river flow and increase in sea level would impact the Franklin County economy, the stakeholders' profession, and the stakeholders' personal interaction with Apalachicola Bay. That could include activities hiking, boating, other engagements such as fishing, birding, hiking, or any engagement with the ecosystem outside of the person's profession. The majority of survey respondents thought a reduction in river flow and increase in sea level would have a negative impact on the Franklin County economy, and then little to no impact on stakeholder profession. Most respondents expected a reduction in river flow to negatively impact their personal interaction with Apalachicola Bay, and that an increase in sea level would have little to no impact on personal interaction. So these results expand upon the impact to Apalachicola Bay species populations by offering a glimpse of how changes in river flow and sea level rise might affect the human dimensions of the Apalachicola Bay system as well. So to sum everything up, we saw that changes in river flow and sea level rise will have varying effects on the Apalachicola Bay food web through changes in environmental conditions, fishery species populations, broader food web dynamics, and impacts to the human community. A notable pattern in the model results was that there were greater differences in environmental conditions and species biomasses between scenarios of low and high river flow than between scenarios of low and high sea level rise, indicating that Apalachicola river flow appears to have a stronger influence on these variables than sea level rise. Temperature was predicted to increase across all future scenarios, while salinity increased during low river flow scenarios and decreased during high river flow scenarios. The food web model showed that future scenarios of river flow and sea level rise would have varying impacts to fishery species populations. During low river flow scenarios, biomasses of commercially important white shrimp and blue crab increased, while biomass of recreationally important Gulf flounder and red drum decreased. Across all, there's an increase in total food web biomass and a decrease in upper trophic level diversity. The stakeholder survey revealed some anticipated negative impacts of sea level rise and reduced river flow to the human community, most notably regarding the Franklin County economy and stakeholders' personal interaction with Apalachicola Bay. So one of the benefits of this study is that the food web model will remain available as a tool to be used by ANERR or other interested parties that can be adapted to incorporate new data and address other questions pertaining to environmental and species population changes in Apalachicola Bay. This study also serves as an example of the comparison of different knowledge types to better understand changes occurring in the Apalachicola Bay system. In some cases, the model and survey results were complementary, while in others, there were differences between the two. And the instances where there are differences can provide areas of further study to better understand the reasoning behind these differences. So analyzing these multiple knowledge sources provides a more nuanced understanding of the system. And this is an approach that can be adapted to assess other climate and human-induced impacts on estuarine systems worldwide. All right, thank you all for listening. I want to give a thank you to everyone at ANERR who's helped me throughout the project. Also a thank you to my collaborators at Tulane who developed the hydrodynamic model, and a thank you to my lab as well. So now I'm happy to take any questions you have.
Okay, thank you, Kira. Looking in the question box, I don't see anything yet, but we'll give people a little bit of time. We're having some bandwidth problems here, so I'm going to keep, but we got a comment. Great job, Kira. So
Thank you.
We do appreciate all your help. Okay. David Adderstein, you want to come off mute? Okay. You should be unmuted, David.
Hear me now?
Yes.
Was I hearing that with low river flows and high salinity, shrimp will get bigger and blue crabs will get bigger?
Yeah, so the results of the food web model showed that the biomass for those did increase in the low river flow scenarios. And that can either mean the individual organisms are getting bigger or they're becoming more abundant. It's sort of a combination of those things, which I know is a little unexpected based on trends that have been seen there in the past. But like I mentioned, the food web model was not able to take into account some outside factors like habitat changes or new predators or overfishing. So adding those additional factors into the food web model for further analysis might give a more informative result. But for what it's showing for now, the biomasses of those species did increase in the low river flow scenarios.
So you're laying a basis, you're laying a groundwork for how to use these tools to get more precise estimations of the effect of these factors?
Really the first time all of the species monitoring data and environmental data have been put together into one tool for the system. So it's really setting the foundation for it to be used for more analyses in the future.
Any observations on the FWC monitoring data in terms of how much was gained from that, how sufficient that information is? I'm just curious about, these are self-reported, that kind of data that you use from the FWC.
So which data are you referring to? The fisheries independent monitoring data or the like fishing fleet landing amounts? Which one were you referring to?
Both of them. And I
Okay.
wanted some interest for you to say what you found in looking at them in terms of how accurate these numbers were.
So in my experience, those tend to be fairly accurate, especially with the fisheries independent monitoring data. They've been collecting their data for 20 years and have really settled in on an efficient collection method and collect it throughout the bay. And for this study, I was looking at both the FWC data and the monitoring data ANERR collects as well. And I've compared those different data sets and they tend to show very similar trends. So I think they're generally pretty reliable. And as for the fishing effort data, those have been collected for several decades as well. So I think those generally tend to be pretty accurate.
And you looked at a broad range of species. You just were talking about shrimp, crab, and flounder, but you had, I guess,
Yeah, so sorry, you couldn't finish.
no, go ahead. I'm
Okay.
just curious about what you looked at or how it would apply to different fish types.
Yeah, so there's about 50 species that are in the food web model. For this project, I just chose to focus on those four, just to look at some of the most commercially and recreationally important. But the food web model does output results for all of the species, so you can really pick and choose what ones you want to look at.
Okay. Interesting. Well,
Thank you, David.
thank you.
And Jenna Harper, you have a question? You want to come off mute?
So beyond red drum, what were some of the other species that the stakeholders were really interested in?
So let me go back to that slide.
Sorry if I missed it.
Oh, whoops, there it is. Okay. So for the commercially and recreationally important species, I had a predefined list that the stakeholders would rank in terms of what was most commercially or recreationally important to least. Then they could also provide write-in answers. Most of the time, there weren't very many write-in answers. Red drum was kind of the one exception to that, where a lot of people included that as a write-in answer. But yeah, so the ones I selected were pretty much the species that people seem to rank consistently as the most commercially or recreationally important.
Gotcha, thank you.
Mm-hmm.
Great. I don't see any more questions. Kira, thank you so much. We really appreciate you coming on today. And for future information, we had a SciCafe scheduled for November, but it's been postponed. Hoping to have Devin Resco from FWC on to talk about the oyster restoration in the bay. We're trying to find a date that works for both of us in November, so keep your ice field. We do have December 15th, our SciCafe is going to be restoring the lateral connectivity of the Apalachicola River floodplain, an adaptive management approach. That's going to be Matt Klondorf, professor at the University of California in Berkeley. So we're looking forward to that. And we thank you all for joining. And again, thank you, Kira, and thank you all for attending. We appreciate it.
Thank you for having me.